All right, well, I'd like to welcome everyone to the first Polyploid webinar here of 2023. And we have two really great talks here on polyploid genomics today. Up first is David Wickell from Cornell University. David received his bachelor's and master's degrees from Wichita State University. Working in James Beck's lab, he found an affinity for seed-free plants, studying the asexual biogeography of Myriopteris grassless, a dry adapted species of fern. He is currently working on his PhD in Fei Wei's lab at Cornell University. At Cornell, David is involved in research on an array of subjects in seed-free plant genomics, from whole genome duplication to the convergent evolution of CAM photosynthesis in aquatic lycophytes, or isoides, as we know them as. With that, I'm going to let David take it away today and talk about his work on diploidization and genomic rearrangement following whole genome duplication in seed-free plants. Thank you, David.
Thanks a lot, Mike. I'm really excited to be here. Thank you, everybody, for coming. Very excited to finally get to present in this Polyploidy webinar. And yeah, today I'm going to talk about diploidization and genomic rearrangement, or a lack thereof, in fact, following WGD in seed-free plants. So, see if I can get my slides to advance. There we go. All right. I am not going to spend a ton of time talking about the process of whole genome duplication, right? Trying to know my audience here, and I assume that you all have this pretty down. But I do want to briefly discuss its sister process, diploidization, right? The process by which we go from this state to this state. Over the last 100 years or so, it's become increasingly apparent that ancient polyploidy is a relatively common feature of multicellular organisms, from plants to insects. And without this process of diploidization, right, we would expect to see a lot of plants and animals that have genomes characterized by high chromosome counts and relatively large proportions of duplicate genes. In addition, polyploidy can result in formation of multivalence at meiosis and disrupt typical patterns of disomic inheritance and expression. And we don't see all that, right? As I'm sure everyone here knows, instead, a lot of species that have a known history of whole genome duplication, like, for instance, Arabidopsis, have very small genomes. They have diploid pairing of chromosomes and disomic inheritance. And this reflects this process of diploidization, which really encompasses two separate processes, though they're tightly coupled, right? That of gene loss or fractionation, and also of chromosomal rearrangement. Both of these processes are known to proceed at different rates, not only between, but also within different species and lineages. That being said, in plants, it typically occurs quite rapidly, has been demonstrated in a variety of groups, right? Take your pick, rice, cotton, maize, tragopogon. And also, these processes of fractionation and chromosomal rearrangement seem to be tightly coupled, with a lot of the genes that are lost during fractionation being lost as a result of processes like illegitimate recombination. So as a result of all this, right, we can have these varying trajectories of diploidization that can have very different outcomes and serve to sort of further isolate these lineages from each other and also from their diploid and polyploid relatives. All right. Unfortunately, studying this process of diploidization in any detail requires a great deal of knowledge, not only about gene and repeat content, but also about the location of those genes and repeats along scaffolds and structural information about those chromosomes that they reside on. While the last decade has seen a serious uptick in the number and quality of plant genomes published, shown here in this little graph, there's been a bias in much of that effort towards species with small genomes and those of economic importance. As you can see from these bars, right, which just show the number of species sequenced in a lineage, we've sequenced lots of flowering plant genomes. And then these numbers just represent sort of the percentage of estimated diversity that we've sequenced. And the reason that there's an asterisk here next to lycophytes is this graph doesn't even really capture the complexity of this bias, right? In lycophytes, I mean, we got 0.3% of diversity sequence. That looks pretty good. But all of those genomes are from heterosporous lineages, and all but one of them is from Selaginella, which, while it is the largest genus, right, it's also sort of unusual among lycophytes in that it has these very small genomes, which is the reason it was the first one we sequenced. So ferns and lycophytes are both known for having very large genome sizes as well as high chromosome counts. This is very well illustrated by this Ophioglossum reticulatum squash. Here's showing 2n equals over 1,200. And so as such, despite their phylogenetic significance, right, as being sister to all of other vascular plants, or ferns being sister to the seed plants, there haven't been a lot of genome assemblies. They've been out of reach of sort of traditional short read sequencing and assembly. Thankfully, right, the improvement in long read sequencing technology, as well as kind of new scaffolding and assembly technology has allowed us to start producing more and more of these kind of large, complex genome assemblies. And in the last year has resulted in 3 homosporous fern genomes being sequenced. This represents a huge opportunity for people who are looking to study both whole genome duplication and the subsequent process of diploidization in these lineages. So I just used a couple of words that I didn't bother to explain, homosporous and heterosporous. I acknowledge that we are not all plant people, so I'm just going to take a moment. If you were confused by that, this slide is for you. So in contrast to seed plants, right, seed plants being gymnosperms and angiosperms and flowering plants that are all heterosporous, seed-free plants can be broadly divided into homosporous and heterosporous groups. Briefly, the heterosporous plants that are highlighted here in blue produce 2 different types of spores: megaspores, which then produce megagametophytes, which then produce archegonia that make eggs, and then microspores, which yield microgametophytes that then go on to produce antheridia that release sperm. This is by far the most common form of reproduction in plants and has evolved independently in ferns, lycophytes, and seed plants. Homosporous plants, on the other hand, produce a single type of spore yielding a bisexual gametophyte that's capable of producing both antheridia as well as archegonia, so sperm and egg. In addition to these obvious differences in life history, genome sizes tend to be much larger and more variable in homosporous groups, shown by this lovely graph from an even lovelier review by Sylvia Kanosian last year, where you can see that these homosporous lineages have genomes that are 3 to 4 times the size of their heterosporous counterparts. There are a couple theories regarding why this might be. I was going to spend time talking about it, but my talk was way over time. So if you want to hear a quick discussion about it, you should go back to Zheng Li's webinar from a couple of years ago on this exact topic, where he does in fact review some of the theories about why we might see these really high chromosome numbers in homosporous lineages. All right, so now I'm going to get on to some of my actual research. Now that we've got the background info out of the way, I want to talk to you first about whole genome duplication in the Syntenic, something that has turned out to be a somewhat controversial topic, and then about collinearity in our Alsophila spinulosa genome. And this will be, intra-specific collinearity in the Alsophila spinulosa genome. And the reason I point that out is after that, I'm going to talk to you about a couple of new genomes. I believe at least one of them is up on phytosome in another lineage, a homosporous lineage, the Lycopodiaceae. I'll talk a little bit about their evolutionary history, and then about both inter and intra-specific collinearity that we found in those genomes. All right, so as I'm sure many people in this webinar know from painful experience, the history of whole genome duplication can be really difficult to determine. And this is in the best of cases, right? When we have all the resources, when we have all the omics available to us, it can still be really difficult. So maybe it's unsurprising that in the Syntenic, a group where we certainly don't have all the omics available to us, that whole genome duplication and its history is sort of a fraught subject. I've illustrated that here with 4 more recent studies, all of which found kind of subtly different hypotheses about where these duplications might be located. I also want to point out, as indicated by the asterisks here on this lineage, that not all of these studies included the same taxa, right? Sampling was very different between some of them. So for instance, these 2 studies found a duplication unique to Alsophila or Alsophila spinulosa, but it was the only member of Syntenic included in those analyses. So had the sampling been similar, right, among all these studies, we might see a little more concordance. Now, I'm not going to spend a lot of time talking about these sort of different hypotheses, except to draw your attention to ours. indicated here in yellow circles. And we figured this out basically by looking at the Alsophilis spinulosa genome. I don't think the book is closed on this, by the way, to be clear, and would be happy to talk to people kind of about some of the controversy. So looking at the genome, right, we kind of did the usual battery of tests. We did KS analysis, which again, it's been explained a lot. I'm not going to explain it here. Suffice to say, we look for these peaks that are indicative of whole genome duplications. If you see peaks in the same place in multiple species, right, we might infer that they share a whole genome duplication sometime in their past. We also did phylogenetic reconciliation using maps developed right in the Barker lab at University of Arizona. And we were able to locate 2 whole genome duplications, one prior to the diversification of Syntenic, and another prior to the diversification of the order Synteniales. And this is all well and good, right? Happy to shed some light on a controversial topic, but what was really eye-catching was this Synteny. So here I have a very beautiful, albeit not that informative, Circos plot, just showing the incredible amount of intragenomic synteny in this genome. And I say incredible because that most recent whole genome duplication, right, that we infer that most of this synteny came from. In fact, I think this particular plot is restricted to gene pairs within the KS peak of this whole genome duplication. We estimate that happened before the divergence of Syntenic, making it at least 100 million years old, meaning that this is unprecedented in plants and extremely rare even in animals, where I think the most commonly cited example is in salmonid fish. So really incredible example. Unfortunately, right, we're going to have to wait for more genomes. I know we're all sick of hearing this from other members of the Synteniales and Synteniaceae to really nail down where these duplications took place and to what degree collinear relationships might be retained in these other tree fern lineages. And the reason that I put this caveat in there is because while it's very tempting to extrapolate this result to all the Mossperous fern lineages, there are a couple issues with that. And they're called Ceratopterus richardii and Adiantum capillus veneris. So I said earlier, right, this was just one of 3 Homosperous fern genomes published in the last year. And while we have this beautiful synteny preserved from a whole genome duplication in Alsophila, we don't see that at all in Ceratopterus or Adiantum. And Ceratopterus, in fact, has a duplication inferred more recent than the one we see in Alsophila. So what the heck's going on? I wish I knew, but I think that, you know, this is sufficient to say, first of all, that there may be different processes, right? There's probably a complex process governing the number of chromosomes that are retained in these Homosperous lineages. And that this sort of link to less recombination or slower recombination might not be as strong as the Alsophila genome kind of leads us to believe, right? There are probably other things at work. All right, so now I'm going to move on to a whole different group of plants, still Homosperous, but these are the plants in the Lycopodiaceae. This is a genus of, or sorry, a family of vascular plants, vascular lycophytes. It is sister to another 2 groups of lycophytes, Isoetaceae and Selaginolaceae. Both of those are the heterosperous lycophytes that I talked about earlier. So this is a partnership with the Chinese Academy of Agricultural Science. They did the Hupertzia genome, and then along with JGI, we sequenced the Diphasiastrum complanatum genome. And before I say anything else, I would like to point out that there are 3, not 2, sort of Circos plots down here. And the reason for that is our collaborators in China were actually able to separate the 2 subgenomes of Hupertzia asiatica, which is a relatively recent allopolyploid. And when I say relatively recent, I mean in the last 30 million years or so. And this was really cool, right? We were able to sort of run these as separate genomes in all of our analyses, which was both informative and considering that Hupertzia asiatica has something on the order of 140 chromosomes. This was much more computationally efficient. So again, usual battery of tests. Here we have rate-adjusted KS plots for one of the subgenomes of Hupertzia asiatica. As we'll see in a minute, the 2 subgenomes are highly similar, and you know, across the board we got the same results for subgenome A or subgenome B. The KS plots in this case were a little bit of a nightmare because we observe very low substitution rates in this group. And along with those low substitution rates, there are independent duplications on almost every single branch in this tree in the Lycopodiaceae. So KS plots are generally kind of a mess. Thankfully, we were able to turn to phylogenetic reconciliation and Synteny, right, to kind of confirm these duplications and try to place them on a phylogeny, where we found one common to the Lycopodioideae in Diphasiastrum complanatum, and then two, one common to all of the genus Hupertia, and then the other one prior to the divergence of our two subgenomes in the Hupertia genome. So while the intraspecific synteny, right, I don't want to gloss over this too much. The intraspecific synteny is still pretty impressive, considering this duplication in Diphasiastrum complanatum is equivalent in age to the one in Cyathea that I was talking about earlier, a little more than 100 million years. But what's really incredible, and you can see in these less pretty but much more informative dot plots, is the interspecific synteny that we see in Lycopodiaceae. So we have this very pretty Circos plot, and this is just showing synteny between Diphasiastrum complanatum and one of the subgenomes of Hupertia asiatica. But next to it, right, again, we were able to treat these subgenomes like separate genomes. And when we compare them, we see that structurally, they are just almost identical. And you know, 30 million years isn't nothing. I mean, this is still quite a while that these two subgenomes have been diverging and yet still display basically chromosome level synteny across the board, with just a couple examples of fissions or fusions or inversions in the entire genome. But then, right, when we compared Diphasiastrum to Hupertia, I mean, these two species are as far diverged as two species within Lycopodiaceae can be, which is a lot. The most recent common ancestor of Lycopodiaceae existed around 400 million years ago. So despite those 400 million years, right, of sort of independent evolution, multiple independent whole genome duplications, we see a lot of synteny preserved between these two genomes. Something on the order of a quarter of the genes in the Diphasiastrum genome have syntenic relationships represented in the Hupertia asiatica subgenomes. And if you look closely, you can even see that some of our scaffolds still have this kind of beautiful like 1-to-4 relationship where almost the entire Diphasiastrum chromosome is present in some form or another on the Hupertia scaffold as well. So yeah, this is really incredible. And it's from two really divergent species, right? Suggesting that this might be a feature common to the whole group, unlike the ferns, where we seem to have a much more complex thing going on. So finally, I just want to point out, we ran a lot of tests that were not just looking for whole genome duplication, right? To keep this short, I've left most of them out, but I do want to briefly mention substitution rates. So we essentially estimated substitution rates, letting branches vary independently in PAML, and then compared them to trees, right, where the entire tree had the same substitution rate, and looked for examples where multiple substitution rates, right, described what we saw better than a single substitution rate. And in almost all cases where that happened, we saw deceleration of substitution rates in Lycopodiaceae. This has also already been demonstrated in Cyatheales, but with even better sampling, right? We were able to sort of reproduce that result in Cyatheales, this lower substitution rate. And, you know, I'm not sure this is directly related anywhere, or if it is, I can't think of how. But that being said, it is a correlation that we see, right, between these two groups that sort of tracks this genomic stasis, this lack of rearrangement that we see in these genomes relative to either their heterosporous, in this case, or homosporous relatives. All right, so I'm pretty short on conclusions here. You know, I'm happy to hear what other people think about this, but basically, you know, I hope that this kind of lays a foundation for people to go on and do more nuanced work in these groups and really try to tease apart what's leading both to the higher chromosome counts generally in homosporous plants, but also, right, this really low rate of rearrangement and recombination in these two groups that we've looked at. Both of them have just extraordinary slow evolutionary rates, both at the level of individual genes and when we look at the rearrangements throughout the genome. So yeah, I think there's a lot more going on here, but I think that this sort of establishes that this is a really interesting system where we have something unique going on that you know, we need to explain. So with that, I want to acknowledge everybody who helped me get here. Everybody in the Lee Lab at the Boyce Thompson Institute on Cornell's campus, my collaborators in China on both the Alsophila genome as well as the Diphasiastrum and Hupertia genomes, Dr. Jim Liebensmack for helping us out with the Diphasiastrum genome, and of course, our very own Dr. Mike Barker and Zheng Li for helping me with all of the maps analyses that I had to do for these projects. And with that, I'm happy to take any questions in the chat.
All right, that was a great talk, David. We have plenty of time for some questions here. So if folks have questions, feel free to put those in the chat or pop on your video, raise your hand, and fire away. And it usually takes a minute.
Fair. Fair.
Well, while we're waiting, I have a really quick question. Do you know the Syntenic depth ratio for the Lycopodiaceae genomes, for the comparison there between Diphasiastrum and Hupertia?
Yeah, it's 2 to 4.
Oh yeah, okay.
So what we would expect, right? With
what you'd expect, yeah.
Yeah.
That's pretty cool.
Yeah, it's very weird, and you know, with both of these, I mean, I feel like I reran these Syntenic analyses like 5 different times because I was just sure I was doing something wrong, you know, especially after doing isoedes, where, you know, again, we have a history of whole genome duplication, but after, you know, 50 to 100 million years, just no synteny in that genome.
Yeah, it's interesting. We've got to get better. Well, as you know, you guys have some of our plants for Selaginella. For some of those Selaginella, we don't have chromosome level assemblies for all those genomes, but it's wacky there. We'll talk some other time. You know, even within, I think in Selaginella, there's this weird thing where the chloroplast genome has lost repair enzymes. And so we see like crazy asymmetric rates in our hybrids and our allopolyploids where there's all this extreme maternal bias in terms of the gene retention and loss in terms of those fractionation patterns, depending on who the parent of origin is, like, you know, 10-20% kind of bias. And I think it's because of that loss of repair enzymes there. And I kind of wonder how much that kind of biology plays a role in other lineages, some of these cytonuclear interactions, as well as, you know, because we often think, and I too think about those substitution rates got to be driving this because it's connected to life history and generation time and all of that, probably, right? At the end of the day. But anyway, yeah, we should talk some other time about this too.
Absolutely. I mean, really briefly, you bring up bias, and you know, that's another really interesting thing that we found in both of these cases is there was no clear expression bias, right?
Subgenome bias.
Either in the subgenomes of Hupertia, which diverged relatively recently, or in Alsophila, we weren't able to find any pattern between either blocks of genes, Syntenic blocks, or whole chromosomes having different expression rates than other blocks of chromosomes. Right. It's interesting.
Yeah, and I know that there's this conflict between these different results, but from different studies about where those duplications go. But you know, the sampling's been different, as well as I kind of also, you know, sort of wonder how much of this may be, you know, we're picking up different signals of hybridization or not too with some of these analyses, because none of us are accounting for, none of these methods account for, you know, ILS or hybridization in those models that everybody's using. We're just looking at duplication and loss, basically. So I do often wonder how much that plays a role, depending on the nature of some of these ancient polyploidies. But it seems like they could be autos then if they're really symmetrical, which would be from first principles, which you might guess, but who knows at the end of the day too. So definitely. Weird things can happen. So all right, there's a question here in the chat from Vanita. Oh, that was to me, I guess. But maybe we could broadly ask how to separate two subgenomes there from an allopolyploid, how that worked. Yeah.
Well, I mean, we have someone who I think is just about to talk about it after me, but they did actually use K-mer analysis to separate those two subgenomes. And then, you know, have subsequently, I think, confirmed it with a couple other things. I didn't do that exact analysis, but I am sure that Adam, right, is coming up to talk and can probably explain very well how that's done. But yeah, K-mer analysis.
Absolutely. Yeah. All right. Are there any other questions for David right now? All right. Well, if not, we'll you can hold on to those till the end, and we can circle back and catch him once Adam's talk is finished. So save those questions now. All right. So, well, thank you, David. We'll let Adam go ahead and put up his slides.
Cool. Can you hear me okay?
All right. Yeah, we got you. All right. Well, I'll go ahead here and get you introduced. So our next speaker is Dr. Adam Session. Adam did his undergraduate at Rutgers and a PhD at Berkeley in molecular biology, where he published the Xenopus laevis genome, one of the first, I think, and possibly, well, not anymore, but one of the first certainly polyploid animal genomes published. And then he was a postdoc at the Joint Genome Institute studying polyploid plants before beginning his lab here at Binghamton University in fall 2021, where he's focusing on polyploid comparative genomics. And he's currently recruiting postdocs and grad students, if anybody is looking to join a lab this coming academic year. And with that, I'm going to let Adam take it away and tell us about his work on K-mer identification of polyploid subgenomes and subchromosomal architecture.
Thank you for allowing me that shameless plug for recruitment. And thank you, everyone, for listening. I probably should have been part of this group for much longer than I've known about it. I didn't find out about it until I started my lab. And so, I'm going to talk today a little bit about the work I've been doing over the past decade or so, looking at polyploid genomes, which started with the Xenopus laevis. Oh, okay. So first, a little bit of an outline of the talk, or hopefully I can have enough time to make it through all of this. First, we're going to talk about what can K-mer distributions tell us about polyploid genomes, then talk a little bit about what transposons tell us about the evolution of higher ploidy organisms. So there, I mean specifically hexaploids and octoploids, things greater than tetraploid. And then talk about some potential applications of this research beyond polyploidy, assuming we get to that part and have enough time. So first, what can K-mer distributions tell us about polyploid genomes? This is something that I started working on during my PhD, working on the laevis genome. And so if you're familiar with the laevis paper, we published this back in 2016, where we were able to actually identify the two subgenomes of laevis using initially a K-mer method that went into the graph that was used to produce the genome. We use MER graphs in order to do this assembly. And so initially, this method was something that was very much married to Miraculous, which was the genome assembler. But part of what I did during my postdoc was really separate out the method so that can be applied to any organism. Now, I also do want to go a little bit more detail into the laevis genome here a bit, because I want to talk about the motivation for why we were looking for these repeats. And so laevis, if you're not familiar, is a case where the two potential progenitor species that gave rise to Xenopus laevis would be extinct today. Nowadays, we know it's an allotetraploid, so it's a little bit weird to talk about, but at the time that I was starting my PhD, we actually didn't know whether or not laevis was an autotetraploid versus an allotetraploid. And so the way of trying to address this question I had during my PhD was, if there were repeats that expanded while the two species were apart in an allotetraploid model. So I should say I'm kind of speeding through this background because I'm assuming most people here kind of understand the difference between auto and allotetraploid. But the main thing here is when you have the two distinct progenitor species in an allotetraploid model, this gives you a chance to have the two sets of chromosomes in different nuclei. When they're present in different nuclei, transposons are able to paint each of them individually. And since there are certain transposons which have no choice but to spread into all sequences in the same nucleus, that means those transposon expansions are essentially just markers that are going to be maintained over time and allow us to talk about subgenomes. And because within an allotetraploid, for the most part, there's going to be no recombination after the hybridization, these ancient markers are going to be maintained today in the genome. This is in contrast to an autotetraploid model, where there's no point here where the two different sets of chromosomes are in different nuclei. So these transposable elements that paint the chromosomes all in the same nuclei have no opportunity to have an asymmetric distribution across the subgenomes of an autotetraploid. Now, without getting into too much detail of methodology, I just want to say that the repeats, even though they're the biological element we're going for, actually working with repeats in very large multicellular genomes can be fairly computationally intensive. And so we use K-mers because having an overrepresentation of a specific K-mer in a chromosome or even a small segment of DNA, the only thing that can really give rise to that are either going to be satellite expansions or transposable element expansions. And so today we're going to be focused for the most part on transposons, but the idea here is that the K-mers are a much more rapid way of studying these repetitive elements. And so the method we use here is we first identify chromosome groups based on shared protein coding genes. So this is just saying that, say we have a base of 3 chromosomes, we don't know what the subgenomes of a polyploid are, but based on shared protein coding genes, we might be able to say within each row here, we know that these two are related versus these two and these two. The next, what we can look for is within each of these rectangles, look for K-mers which are differential between the two groups. So this literally means just counting the K-mers in your 2 chromosomes and finding things at this point which have a ratio difference between them. In these early steps of the method, we actually just use a ratio of 2x. With the current version of the method, we're currently putting out a paper that's about to be published, and the associated GitHub allows you to choose what that initial ratio you want to look for is. But at the end of the day, we just have 2 chromosomes and we find K-mers that are overrepresented in one compared to the other. Then the next step is then to look across the different groups and find a group of K-mers, which is the intersection of all the diagnostic K-mer, intersection across all groups. And so these K-mers that are across all groups represent transposable elements which expanded while the 2 species were apart and are still maintained today. So overall, what this looks like is being shown by the heat map here. So the individual rows, even though you can't see them, are actually K-mers. And so in this case, this is using a K equals 13. And then the columns here are the different chromosomes of Xenopus. And so we can find K-mers that are enriched in what we call the L subgenome, and another set of K-mers which we call enriched in the S subgenome. The L and S here stands for long and short, which are the physical properties of the chromosome, which I won't get into too much today because that kind of deals with the bias fractionation side. I just want to focus on the identification of the subgenomes today. So the way this looks in terms of a statistical validation, though, is that we can do an ANOVA, where the groups that we're comparing are the L subgenome and the S subgenome. And we're asking how well each K-mer is able to differentiate between those 2 groups. And so on the x-axis here, we're looking at the mean S minus L K-mer count. And then the y-axis is negative log 10 of the Tukey p-value. And so basically, it's just a measure of how well each individual K-mer is able to differentiate the L and the S subgenomes. So each dot here is a 13-mer that we're actually looking at. The red and the blue coloring here are things that were in that initial ratio analysis, things that we found to be differential across all of the pairs of chromosomes. The gray line is really just the result of if you were to do a Bonferroni correction in order, because we're looking at millions of pairs here, then it's a very aggressive way of filtering this data. But we'll come back to this, assuming we have time near the end of the talk. But the main thing here is that we're able to take these K-mers, you align them to the genome, they overlap with known repetitive elements. And so in the case of Xenopus, we're able to find multiple families, one of which was this Harbinger subfamily, which was an ancient Xenopus subfamily that had unique expansions in the L and the S progenitor species. But specifically, the Mariner subfamily was the most interesting because it was something that was exclusive to S. It gave us a roughly 2 kb consensus sequence that would only be on one set of chromosomes. And we can probe the chromosomes of Xenopus with this because there's so much cytogenetic data already existing for Xenopus laevis. And hopefully what's clear here is that there's a lot more of the Mariner sequence on the S chromosome. It's almost non-existent on the L chromosomes. This type of pattern where you get this repeat overrepresented on S compared to L can only happen in an allotetraploid model because there's no recombination going on between the L and the S progenitor or L and S chromosomes here. So these ancient transposon expansions are maintained today. So I worked on a number of different plants in my postdoc. Today, I just want to only focus on Miscanthus sinensis because it's a unique case where, we did the exact same algorithm using the K-mers where we identify A and B subgenomes. And then we're able to look at the entire Miscanthus genome compared to the diploid sorghum. Now there's, this chromosome fusion, which you may notice, but there's some other kind of things here which I want to zoom in on and focus on. And specifically, it's looking at this chromosome 5-6 pair and chromosome 11-12 pair. Now, these plots are a little difficult to interpret, and I eventually move on to a better visualization. So apologies for this old version of looking at this. But the general idea here is that the density of these dots represents unique instances of those unique 13-mers that are subgenome specific. So you have more blue on the top row because that's the A subgenome, more red on the bottom row because it's the B subgenome. I'm sorry, I said subgenome specific, but I should say enriched, mainly because we don't demand exclusivity here. Now, the unique thing here is that these terminal ends of the chromosome appear to swap the subgenome type in miscanthus. And these are things that we're able to confirm through physical mapping of these sequences. And then this is something where if you look at the bias fractionation of miscanthus, that seems to get disrupted in this region as well. Additionally, when we look at other polyploid miscanthus species, so not Syntenic, we find this exact same swap between the subgenome types at the terminal ends of these singular chromosomes. And so this is what we expect to find if we were to actually observe homologous recombination in a polyploid species after the hybridization event. Or at least currently, this is what we are able to find in terms of the genomes that we've worked on so far. And so the actual assignment of these to the opposite subgenome, I won't get into the details of, but we use a hidden Markov model if you're interested in that. And if you want the details of that, please, ask me questions. I'd love to talk about it. I just wanted to get this talk down to 20 minutes. So next, what can transposons tell us about the evolution of higher ploidy organisms? So for this, I want to use strawberry as an example. For what some people may not be familiar with, is strawberry is an allo-octoploid. And there's a wide radiation of diploid, tetraploid, hexaploid strawberries which are studied. However, in the initial analysis of the strawberry genome, subgenomes were called based on alignment to the based on sort of looking at the closest diploid relative species. However, in general, this method does demand that you've sampled the actual diploid progenitor of each of the subgenomes you want to look at. So when I wanted to try my method on a higher ploidy organism, so we came to strawberry. And specifically what we find is we actually agree on two of the subgenomes. So specifically what are called the Vesca and Anume subgenomes being shown on the bottom here. These are things that are closest to the diploid Vesca and diploid Anume. And we find K-mers that are exclusive to those two subgenomes and actually shared with the diploid species that they've been shown to be related to. However, the other two subgenomes we actually disagree on. And the pattern of K-mers there is not necessarily straightforward, and so I just want to take a second to explain it. One of the subgenomes that we're calling alpha has its own set of exclusive K-mers. We have another set of K-mers which are shared between alpha and what we call beta subgenome. And then finally a set which is shared between the Anume, alpha, and beta subgenomes. We'll talk more about what these shared signals mean, but the main thing I want to say here is we find unique sequences to V, I, and alpha. And beta can be defined by the presence of the alpha beta, the I alpha beta, but no alpha K-mers. So again, we find these subgenomes which differ from what has been proposed previously by Edger 2019. And so in order to actually try to statistically validate which one of these may be better supported by the data, again, we go into an ANOVA plus Tukey's HSD test in order to ask how well each of these clusterings is supported by the K-mers in the genome. And so again, we are looking at volcano plots here where it's mean, in this case, Vesca minus Anume, Anume minus alpha, K-mer difference on the x-axis. Y-axis is negative log 10 Tukey p-value. And using the Vesca and Anume as sort of an example, the red K-mers here are the V K-mers. Those you can see are higher in Vesca. The Anume and I alpha beta K-mers are higher in Anume. And the things that separate alpha and alpha beta from the others are not significant when comparing Vesca and Anume, as you might expect. And you find a similar pattern here when comparing Anume and alpha. However, kind of the more interesting thing would be to ask about the beta alpha versus Japonica viridis clusterings. And when we look at beta minus alpha K-mer difference, we find a strong group of K-mers which are higher in the alpha subgenome compared to beta. While there are things higher in beta compared to alpha, these do not form kind of consistent groups which align to specific families of repeats, which we can then do phylogeny on. So there may have been in the past a unique expansion to the beta subgenome, but that sequence would have just been lost today. But we can't actually separate that from the idea that maybe the beta species just did not have its own unique transposon expansion. I won't get too much into the timing today, but we believe that the alpha and beta species only existed as diploids for roughly 1 million years. Now, the other side of this would be if you did the Japonica viridis clustering and you asked how well each K-mer can separate those two subgenomes, you find that no K-mers actually separate those subgenomes. The reason for that is because the Japonica viridis clustering is actually just randomly putting together the alpha and beta subgenomes. And so there will be no reason that there will be K-mers which separate the Japonica viridis clustering because it is essentially a random assignment of the alpha and the beta subgenomes. So if you're curious about how these K-mers look distributed along each of the chromosomes, this is organized into Vesca, Inume, alpha, beta subgenomes. Then we're looking at the Vesca-specific sequence on top in red. You can find it's mostly, and this is a histogram looking at 100 kb bends, so the number of base pairs per 100 kb. And you can see that the Vesca signal is specific to Vesca. We have the hexaploid signal that's not very high in Vesca, or not very high here, and then similarly for the others. So these seem to be fairly clean signals with no evidence of recombination between the between the 4 subgenomes. Similarly, I talked a little bit about what the shared signals mean. I won't get into too much detail, but essentially here, the alpha beta shared signal is evidence of a tetraploid ancestor between the alpha and beta subgenomes. The I alpha beta signal is the evidence of an hexaploid ancestor between the Inume alpha and beta subgenomes. And so this K-mer identity not only helps you understand the subgenomes and assign them to specific groups, but in case of hexaploid, octaploid, and these higher ploidy organisms, it also allows you to understand what order were these diploid genomes hybridized into a single organism. And so this is currently on press. We've also done this method in a number of other species, including hexaploid camelina. And so if you're interested in those results, I mean, this is something that should be coming out very soon. We're in the kind of the final stages of review at Nature Genetics. And so finally, I'm going to spend just a very short amount of time talking about what are some potential applications of this beyond polyploidy. And one of the things here is that even though I'm kind of going beyond polyploids, what I'm hoping to do with this research is really establish polyploids as a sort of model system to study genome evolution. And what I'm hoping to do in this last section is convince you that's, pretty exciting. Not that I need to convince this group of that, but so returning to that thing I showed earlier, what I didn't say is that this version of this figure is actually updating my analysis to the new version of the Xenopus laevis genome. So back in 2016, we were working on a shotgun plus genetic map assembly. We did some higher order contact map, but it was not a PacBio assembly. The new one is we have a much better repeat landscape in this new assembly. And so specifically, I want to come back here and talk about what are these outliers here, which have a mean K-mer count that is, hundreds more than we find in L. When we look at these K-mers and we map them to the genome, what we actually find is that they build up in the terminal ends of the chromosomes. And that's because these K-mers are actually markers of satellite expansion specific to the subtelomere. If you're not familiar with the general geography of a chromosome, the terminal ends of the chromosome being shown in red here, you are probably familiar, are called telomeres. The yellow here is a centromere. But there's actually a repeat-rich region called the subtelomere, which is adjacent to the telomere, but is rich in repeats that are not just the telomeric repeat, just to make that clear. And that's being shown in light blue here. And so what we're actually identifying within this analysis is the subtelomeric sequence that is specific to the S chromosomes within Xenopus. And so the general method that we're using here is to trim off the terminal ends of the chromosome. I will say this 20 megabase length is something that's specific to amphibians. The subtelomere length in mammals, for example, is around 500 kb to a megabase. And so they don't pay too much attention to this length here. It's something that changes very much even within vertebrates when comparing different groups. But the idea is still going to be very similar to what we talked about earlier, where you're going to trim off the ends, count the K-mers within each group. So we have subtelomeres versus interchromosomal sequence. We're really just looking for enrichment, which we can judge by ANOVA and Tukey's HSD. And then finally, we can use a hidden Markov model to try to map the boundaries of the subtelomeres. The end result of that hidden Markov model is shown here. And I know this isn't perfect, it's still an ongoing analysis, but I do think that this is actually pretty promising overall. So we do get, I will say one thing here is that we find both L and S specific subtelomeric sequence and subtelomeric sequence which is shared between the two subgenomes. So one thing that's pretty exciting about this result is the fact that within mammals, it was actually hypothesized that perhaps these subtelomeric sequences, the way that all the chromosomes were sharing DNA would be maybe if there was a small amount of recombination going on between non-homologous sequences where these subtelomeres are being exchanged between chromosomes. However, what Xenopus tells us is that cannot be the case, at least within amphibians, because we maintain these ancient subtelomeres. And I didn't talk too much about it, but the hybridization in Xenopus happened 20 million years ago. And so these are sequences which have been maintained in the genome for 20 million years. And the more recent shared subtelomeric sequence, or the shared subtelomeric sequence is more recent. But the other cool thing here is that some of these areas where you actually predict subtelomere in the middle of the chromosome are cases where we actually know that there are genomic rearrangements involving the subtelomeric sequence. So for a very basic chromosome, is being shown by chromosome 1 here. And basically this karyogram is from the 2016 Lavis paper. And the blue lines are connections between the polyploid Xenopus laevis and Xenopus tropicalis. These are proteins that they're, cDNAs that they're probing the chromosomes with. And then also probing the polyploid chromosomes here. I show chromosome 1 as an example of what this typically looks like, where you have the middle of the sequence be solid or inner chromosome, and the subtelomere is called near the end. But for chromosome 4L and 8S, these are two cases where we actually identify subtelomeric sequence in the middle of the chromosome. And these are both, as I said earlier, cases where there's genomic rearrangements which are known to involve the subtelomere, and there's a reason that sequence was brought into the middle of the chromosome. And so we're currently working and also looking at the decay of the subtelomeric repeats when they occur in the middle of the chromosome, hopefully as a way to be able to actually time when these rearrangements occurred. And so instead of having to rely on, chromosome rearrangements happening sort of as speciation events in order to put an exact time, we can start using the evolution of the repeats at the chromosome rearrangement in order to really understand when that occurred. And so even though that's kind of a more general idea, the point of all of this is still that polyploids are going to be a unique natural experiment in genome evolution. And really what I'm hoping my lab can do in the future, which is what the subtelomere comparison between polyploid Xenopus and other tetrapods is about, is really about trying to establish polyploids as a model to study genome evolution. We have a lot of ideas and hypotheses about what happens during speciation and what type of genomic changes happen, immediately following. But polyploids are this experiment where you bring allopolyploids, I should say, where you bring two different species together into the same nucleus. And we can then study how they evolve specifically in order to refine our models about how genomes evolve sort of between species as a whole. And so with that, I want to thank everyone at Berkeley who worked on a lot of this work with my PhD, the entire Xenopus community, the groups at JGI and Hudson Alpha, and still just starting my group at Binghamton. So I'm sorry I didn't credit any of my students now because they haven't made any data yet, but they're great students. And thank you. I'll take questions.
All right. Well, thank you very much, Adam. That was a great talk. Yeah, like last time, if there are any questions, feel free to post those in the chat or feel free to pop on your video and ask away. And I'll hold off on asking my question. All right, I'm going to go ahead and shoot my question in there. So that was really cool. So for the thinking about using K-mers in general for this sort of problem, for example, for identifying rearrangements and like fusions, for example, with the subtelomeres, it seems like it's dependent on, sort of how fast those sequences will degrade. Do you have a sense of, so in Xenopus, that seems like it's fairly slow. Do you have a sense of that from for example, from plants or whatnot? I know that, the LCR, like half-lives of TEs and stuff are pretty young, right? So I'd be curious how,
like this K-mer method to identify subgenomes, I didn't talk too much about it, but like I've done it on a number of Brassica species. So Brassica rapa is the hexaploid that's older than 6 million, if I remember correctly. And that's roughly around the half-life of a TE within the plant genome. And for that, we are not able to actually find any signal in the K-mer data.
Right. And that's about, that thing's actually ends up being about 20 million years or so with our most recent dating. So it's not too surprising. Yeah.
I will say, we are able to find things in strawberry. That base in strawberry, I think, is around 8 million years, if I remember correctly. But within, we know in general, animal genomes evolve more slowly than plant genomes in this factor in terms of how quickly do you clear out repeats or transposons. And so for the oldest burst that we can identify in Lavis is, almost 40 million years old.
Wow, yeah, that's cool.
And so, and that's something that's, specific to one of the subgenomes. And calibrating the times in that requires you to find groups which are shared between the polyploid and the diploid. And you can do that. And it's a lot easier within plants because retrotransposons make this a lot easier than DNA.
Yeah, sure. Absolutely.
You can still kind of go into the phylogeny. There's a lot of kind of tweaking of, you can't use standard math mainly because there's a lot of missing data when you're talking about that. Whereas, with a retrotransposon, you can kind of look at the general, like as long as I can find the long terminal repeat and understand, I can get a pretty good grasp on when that originated and when the
Right.
from looking at all the long terminal repeats. Whereas a DNA transposon is a much larger space that you need to maintain, and
Right.
that still is able to drop out. But
we have pretty good. You should be able to go back in time pretty deep then for a lot of these plant lineages, like that aren't flowering at least. So gymnosperms and ferns have 40 and sort of like, I think we estimated them at like 15 million year half-life. So that's cool.
And so, yeah, it's pretty, I mean, I will say for the polyploidy, it's pretty much how long the transposon lasts.
Right.
K-mers, in terms of the subtelomere and the general rearrangements, that's a bit more complicated because also satellites will have different decay rates than, say, actual transposable elements. And not to mention that, I didn't really get into it, but we also look at the subtelomeres of the Diploid tropicalis. And in that case, it's a satellite expansion that uses sequence from a transposon, but then that transposon gets silenced throughout the rest of the throughout the rest of the tropicalis genome. And so that's something we're kind of trying to understand is, are you truly randomly picking sequences for this satellite repeat within the subtelomere? Or maybe for you to choose existing transposons and then silence them.
Well, that's cool. Yeah.
I will say that's very much a very new result, and we don't understand fully how
we go there. Well, I was asking about this, and I'll stop here in a second, but we might be in touch. We have a new genome from a 2 chromosome plant, an N=2 plant that we know got there through a bunch of fusions. And so we've been just starting to look at analyses to identify where those fusions have occurred. So from the de novo, from the sequence itself, it's all long read data. So we might be in touch to try to see if we can apply this approach to identify those places. It might be really fun. So
yeah, and I mean, this gets at also the question that got posted just now. I would recommend my protocol. I will say my GitHub, I need to update to the latest version after reviewer comments, just to make sure that I'm trying to make it as user-friendly as possible, mainly because I don't want to have to maintain it and answer questions about it. But the general idea, I also just kind of have the general code there, like in terms of if you want to use other programs. But I mean, there's a number of different people who have even talked in the seminar series about their K-mer methods or other methods to identify subgenomes. And so I would also just suggest looking at their methods as well, to not only talk about my own. I mean, everyone else, I'm sorry, I'm forgetting names, but there were multiple people last year who I think gave great talks on, their versions of the same thing. And that all seemed very reasonable. And so in just kind of answering Pepe's question, perhaps too much, but I would say the seminar series is probably your best. And then looking at people's talks is probably the best way of trying to find a good one.
Yeah, and for those of you who aren't seeing the question, Pepe asked both David and Adam, could you recommend any protocol or tutorial about separating subgenomes and chromosomal rearrangements? Just so we're all on the same page there. Yeah, there were a couple of talks last year from folks talking about subgenome recognition. I know Pilar Catalan from the University of Zaragoza talked a little bit about that with their method for finding these ghost subgenomes in Brachypodium, and that's a pretty sophisticated pipeline, I think.
Brachypodium
using
genomes I worked on also.
yeah.
Yeah, I mean, and Pilar, I mean, I would say it also depends. I mean, if you have the diploid, like my method is very much from an idea of, I don't want to have to look at any diploid. I want to try to come up with a way of only looking at the polyploid and answering this. But if you do have the related diploids, the protein coding gene comparison, assuming that incomplete lineage sorting is not an issue, like that is a very powerful way of doing the assignment. But you would just need to also make sure the idea there is that, there should be blocks of genes which are assigned to one diploid at the very least. Or you can think about in the tobacco genome where they take the reads of the diploids and you align that to the tetraploid. I'm sorry, I thought about this a lot, so I can think of a lot of references, and I'm probably just
And I'll say that we have a new paper coming out in genetics using a population genetic approach to do this as well, where we do subgenome assignment while inferring demography in polyploids. And And so I think it really depends on the kinds of data you have too, and how deep those phylogenies are. I think there are sort of different approaches. Just to speak generally about that subgenome assignment. Yeah, David, did you want to say anything? I saw you pop on.
Oh, I just wanted to mention that I have no experience with it myself, but I believe our collaborators used a program called subphaser to separate the two Herperzia subgenomes. If you're interested in that.
That's right, and that's a cool paper that came out I think last year, right? And a tool that was published I think in New Phytologist last year, which is it's on my list. I haven't played with that one yet, but very cool. Oh, subphaser. There you go. If you got, all right. I see Justin has his hand up, and he's been eagerly waiting, I think, for a while. So,
I have Adam. Thanks for a great talk. I did have two questions. Luckily, Mike took my first question about the decay of these K-mers, but I was really interested in your strawberry data, where you showed that there were some K-mers that were specific to the hexaploid and tetraploid intermediates. I wonder if you can use those K-mer data to time when those individual polyploid events happened or not.
Yeah, we did do, so that's in the paper. Again, this is kind of, I was trying to get into 20 minutes, but yes, we have done that. I want to say that's even in the bioarchive from 2020. But basically from there, they overlap with retrotransposons. Retrotransposons, the long terminal repeat must be identical at the time of insertion. So if you then also compare sort of to get the calibration rate, which ended up being the same as May's mutation rate, but we kind of want, we tend to try to calibrate towards the organism we're working on. And so essentially there to try to get the rate of substitution that's expected, we compared the Vesca to the Inume, the diploid transposons, and found the distance there and figured that was the base of the strawberry radiation, and everything's calibrated towards that rate. But it ended up being basically the same as the Mon-Bennetton rate. We just kind of estimated it from the data within the strawberry genome. But you're right that, yeah, if you can find those, you find the time, you can not only find a time when those specific families expanded, you can also see that around 1 million years ago is when all four, the shared K-mer activity between all four subgenomes starts again. And so I will say that my method tends to, once you get under 1 million years, it just tends to say this is under 1 million years and there's no good accurate measurement there. And so the idea would be that the octoploid probably formed less than 1 million years ago or around 1 million years ago, at least from the estimates based on the shared transposon activity that picks up again. Does that answer your question?
And yeah, it does. Does it matter does it matter the source of where the transposon that increases in frequency comes from? Does it have to be present, if you're looking at the hexaploid, does it have to be present in the tetraploid first, or could it be introduced by a new diploid?
They can, you would almost prefer, they could, so it could be that it's present in the tetraploid and expands again, or it could be present in the diploid and expands again, or it can be completely novel, like the Mariner one for or Xenopus, the S subgenome. I didn't really get too much into it, but this does get, you asked a very good question, which is, how do these different types of families respond differently to this method? And so, for example, when you do this on cotton, almost all the repeats in cotton seem to have existed prior to the speciation of the diploids. And they're all very asymmetric, where it's something where it's, basically they're very dense across the entire genome, but you're able to pick up on sort of a ratio difference of 1.2. Whereas here, most of these are going to be most of the ones I talked about, which are sort of, when I did that very restrictive filtering of the K-mers, those are for the most part going to be things which are purely exclusive, like the Mariner. The things that were showing up in the ratio data, but I filtered out, those tend to be things that either existed in the ancestor and had a burst in only one, or unique bursts in both. And so the K-mers tend to be asymmetric, or they burst in one ancestor, but continue their activity in the polyploid. So again, that gives you the same issue where it's present across the genome, but it's just asymmetric. And so you really have to try to look at the origin to understand the timing of that. And I will say, the origin time that we get is by looking at the entire LTR subfamily and asking what is the coalescent time sort of at the beginning, which has its own issues. I'm just trying to be candid about what the methods are doing. Maybe that's way too much information to spit at you, but
not at all. I really enjoyed it. Thanks for being so thorough in your answer. I appreciate it.
All right. Are there any other questions for either David or Adam today? If not, all right, I think we'll go ahead and wrap up. And I want to just thank both David and Adam for two really great talks. I'm going to be following up with both of you about different but still related things. And hopefully we can connect on a couple different things that we got going on as well. It'd be fun to interact on. Those are two fantastic talks, and I appreciate both of you taking the time to do that, especially here right after the holidays as we kick off 2023. And we'll be back here again in about a month. Just want to highlight our next talk. It's 3 weeks, I guess, on February 1st. We'll have Elise Perry come to talk to us about her work on genome evolution in the fishes. So maybe a little bit, if you squint, a little bit reminiscent of some of those really conserved Syntenic plots that David was showing, which fern and fishes seem to have, we got genomes, lots of interesting interactions there. I think there's some fun stuff to do comparing those at the end of the day, but with whatever biology is driving all this interesting diploidization patterns. So we'll have Elise here. We can ask her about that maybe if we all come back. As well as Ben Blonder, who will talk about his work on why there are so many triploid quaking aspen clones, which if you're out west, you can't miss them. And so it should be a lot of fun. And we'll see everyone back here on February 1st. Keep an eye out on for those posts on Mastodon now, since we've mostly moved there from Twitter, as well as emails about the next webinar series. But again, thank you to both Adam and David, and hope everyone has a great start to their spring semester.
Thanks, Mike.
Thank you, everyone.